can start, yeah. Is it going to do an introduction?
Okay, so welcome everyone to our last meeting in our series on philosophy of molecular and developmental biology, and it's my pleasure to welcome Ivan Kuzin, who is a philosopher and biologist, who is now based in the University of Linz.
Thank you. I'm very grateful for the opportunity to speak here. Let me present my presentation, the concept of information and new phenomena in molecular biology. First, I will describe some philosophical discussions on the concept of information in molecular biology. Then I will turn to new phenomena in molecular biology. Then I will try to show that this new phenomena problematize the concept of semantic information, and will outline some approaches to understand new phenomena in the context of the concept of information. And my methodology is integration of, it's not a pure philosophy of science, it's integration of philosophy of science and history of science in order to enable their mutual critique. But this total integration approach requires much time, so I will be very brief at many moments, and if you would have questions, then you could ask them after the presentation. So, philosophical discussions. My initial motivation long ago was to understand what is the paradigm or episteme of molecular biology, because it's a very famous, probably the most famous notion in philosophy of science, and how it is applied to molecular biology. I can't understand. As a description of this, as a source for discussion of the paradigm of molecular biology, we could use the classical textbook, Molecular Biology of the Cell, by Alberts and co-authors. I will use the last edition, and I will use many pictures from this textbook. And one of the possible answers, the cell is a machine. That is a paradigm of molecular biology, and there is a well-known paper by Daniel Nicholson where he argues that it is not true according to new data. And my approach is similar, but I will highlight another group of concepts, informational and linguistic concepts, because they are also embedded in the discourse of molecular biology. And I will use three examples: so-called central dogma of molecular biology, including the genetic code, notion that genes contain information about the phenotype, about the organism or the cell, and the notion of intracellular signaling associated with environmental information and its flow inside the cell. According to the central dogma of molecular biology, genetic information flows from DNA to RNA in a process called transcription, and from RNA to protein in a process called translation. And we could already see that here linguistic notions are used, and linguistic notions I will understand as a subset of informational notions. And the part of the central dogma, the part which explains how explains translation, is so-called genetic code. Code is also informational concept. It's a correspondence between triplets of nucleotides in messenger RNA. Messenger is also informational concept, and amino acids in proteins. Here is visualization of genetic code. We could see that it is looks very natural for biologists to describe relationship between RNA and proteins using letters and using other linguistic and informational notions. More general concept of molecular biology is that genotype contains information about the phenotype, for example, instructions for its construction. Often these instructions are called the genetic program, and indeed, in some models it is useful to interpret regulatory processes inside the cell, as, for example, Boolean networks use logic gates describing transcription and so on. And the third example is intracellular signaling, which intersects with gene expression, with the relationship between genotypes, between genes and phenotype, because flow of information from signal molecule could alter gene expression, could alter relationship between gene and phenotype. So I will discuss them together. The relationship between genes and phenotype and intracellular signaling, because they intersect. And I will discuss information, and I will call biological information only information in an intracellular or intercellular sometimes context, not in a cognitive context, like in animal communication. And I will follow in this and many other aspects to Godfrey Smith and Sterelny in their article about biological information in the Stanford Encyclopedia of Philosophy. When the use of these concepts becomes problematic, when they are understood in an objective sense, ontologically, interpreted ontologically, explanatory, and when they are understood literally. Other approaches are not so problematic. So if we really think that there is information processing, there are information processes inside the cell, how could we define information? I will simplify this question like many others. The minimal notion is the correlational or syntactic notion of information by Shannon. According to this notion, anything could be a source of information. If it has a number of alternative states that could be realized on a particular occasion, and any other variable contains some information about the source, could be considered as a signal or as a message, if its state is correlated with the state of the source. And which is important, it is a matter of degree. It could be quantified. A signal carries more information about the source if its state is a better predictor of the states of the source, and less information if it is a worse predictor. This correlational or syntactic or mathematical notion of information, unfortunately, does not meet the requirements arising from biological practice. I mean asymmetry. In case of a correlational concept, we could interpret signal as a source and source as a signal. It doesn't. It's not important. But in case of biology, biologists say that genes contain information about building phenotype, not about phenotype containing information about building genes. They talk about normativity. There could be mistakes in DNA, there could be mutations that lead to aberrant phenotypes, and according to a minimal conception, almost anything could be a source of information or a signal. But in biology, this word information is reserved for special cases. For example, many biologists and philosophers of biology think that only genes could be carriers of information, not, for example, environment. So correlational information does not meet this requirement, but semantic information satisfies this requirement. Semantic information is modeled on a language, on philosophy of mind. There is a special relationship between word and an object. Word, in some sense, is directed to an object, and it's a special semantic relationship. And some philosophers think that between genes and, for example, phenotype, there is also this special kind of relationship. So at the core of the semantic information is a concept of intentionality, but it needs to be naturalized. How it is realized. And usually it is naturalized using the concept of biological function, because the function is also asymmetrical. Legs for walking is not for legs. Function is normative. There could be malfunctions, and function is not ubiquitous. Not everything in the organism is functional, and if it is functional, it's not functional about everything. So the concept of function is also symmetrical, normative, and non-ubiquitous. Moreover, it could be naturalized using so-called etiological concept of function, which means that function derives from a history of natural selection. For example, the function of a cat is to pump blood, not to make thumping sounds, because it is the former effect that has led to cats being favored by natural selection. So this mysterious intentionality could be explained by a history, by an evolutionary history, by changes in frequencies of genotypes and phenotypes during evolution. But a hat does not contain instructions or descriptions, contrary to genes, supposedly. So this teleosemantic interpretation of information through appellation to function, telos, requires some additional properties, and the most simple alternatives is a presence of a sender and receiver of information, of a signal, of a message, or arbitrariness of relation of correspondence between an alleged signal and an alleged source of information. And Both properties are problematic, which I will briefly discuss. Sender and receiver of message. In case of genetic messages, the sender could be, as the sender could be considered natural selection, and as the receiver, the developmental system of the organism. Authors that like to think that there could be also environmental information, they could abandon the concept of a sender and only use the concept of a receiver. And this concept is better defined at the cellular level, for example, as the machinery of transcription, of synthesis, of RNA synthesis, and machinery of translation, of protein synthesis, or receptors as receivers of information. But on the level of the organism, there are no centralized readers, there are no central mechanisms for controlling ontogenesis. Another alternative is an arbitrariness of a correspondence between analogy signal and analogy source. For example, there is no reason why a word cat symbolizes a cat. We could change cat and a dog. These words we could change them, and it looks like nothing would be broken. And some authors think that the correspondence between signals and sources, like in this example, are semantical because of their arbitrariness. I will look closer at one example, at the genetic code. First, I will contrast it with the process of replication and transcription. During replication and transcription, nucleotides on different chains are paired, and it is not arbitrary, it is stereospecific. But the correspondence between nucleotides of messenger RNA and amino acids is, in a sense, arbitrary. So, correspondence between amino acids to the left and between triplets of nucleotides on mRNA is arbitrary because there are two adapters that connect amino acids and triplet of nucleotides. It namely transport RNA and tRNA synthetase. And changing these adapters, we could change the genetic code. It is experimentally shown. In case of correspondence between genes and phenotypes, or between environmental signals and phenotypes, the principle is the same. The mechanism of arbitrariness is modularity. For example, separation between regulatory and functional parts of a protein. This modularity makes possible independent evolution of different chains of a causal, different links of causal chain. So, in a sense, it is expected, but it looks that any causal correspondence that includes 3 or 4 links will look arbitrary. It's a problem. So, my intermediate conclusion: information concepts are embedded in the discourse of molecular biology. The most obvious way to clarify them is by means of telesemantic interpretation, but the related concepts of sender, receiver, and the arbitrariness of the correspondence require clarification. Now I will go to new phenomena in molecular biology. If we compare new and old editions of molecular biology of the cell, then we will see a big difference. The new editions include discussion of so-called biomolecular condensate. They could be contrasted to macromolecular machines and membrane-enclosed compartments. An example of macromolecular machine is ribosome, a machine that is synthesizing proteins. An example of membrane-enclosed compartment is, for example, mitochondria, lysosome, etc. And these new entities, biomolecular condensate, they are not fixed like macromolecular machine. They don't have fixed composition. They are dynamic. They are often liquid-like or gel-like. They are permeable to small molecules, in contrast to membrane-enclosed compartments. Their stoichiometry is usually not fixed, and they could easily assemble and disassemble. And this new phenomena looks surprising because until recently, very influential idea was that the cell is a collection of machines, and Bruce Alberts, who is the main author of the textbook, was very fond of this idea. Research of biomolecular condensate is new, and the number of papers is increasing. Here you could see the number of papers dedicated to liquid-liquid phase separation. It's a mechanism by which biomolecular condensate could be made. And research on membraneless organelles, it's another word for biomolecular condensate. Information and maintenance of biomolecular condensate usually are involved intrinsically disordered protein regions. It's the second phenomenon that I would like to discuss. Here they are called unstructured structured proteins. Their importance was recognized a little bit earlier, around to year 2000. They lack a stable three-dimensional structure, so they contradict sequence structure function paradigm, according to which function of a protein is determined by its unique three-dimensional conformation, three-dimensional structure, and this three-dimensional structure is determined by its sequence. And here, this determination is much weaker, and they are typical for eukaryotes, and they are very ubiquitous. For example, 70% of proteins in the human genome have IDRs of some significant length. Even when bound, IDRs do not intrinsically disordered regions do not necessarily adopt a stable conformation. They could fold after binding, but they could remain in a number of conformation or even in a completely disordered state, which does not prevent binding. This is surprising. And their molecular function is not determined by a unique three-dimensional structure. Instead, it is determined by a combination of some short linear motifs, some short stretches of amino acids, so-called SLIMs, short linear motifs, by properties of an ensemble as the whole, and by sequence features, for example, how many hydrophobic amino acids are present in this protein, how these hydrophobic acids are clustered, or if they are uniformly distributed. And there are some, I will a little bit clarify. Properties of an ensemble, for example, radius of duration, which characterizes how compact is the protein. They are described by probability distribution. Properties of an idea ensemble are highly context dependent. For example, in case of C, crowding agents, molecules that just make cell look crowded because there are many of them. These crowding, so-called crowding agents, they could make an idea, they could extend idea if they are small, and they could, on the contrary, compactify idea if these crowding agents are big. Ideas are also sensitive to the salt concentration, to pH, to temperature, and to the modification of the protein by phosphorylation, arginine methylation, and many other modifications. Short linear motifs are often involved in the molecular recognition of idea ligands, but their binding also depends on the content, on the context. For example, if in idea there are negatively charged amino acids around SLIM, and in ligand there are positively charged amino acids, the binding will be stronger. So a few words about biomolecular condensates. They often arise from liquid phase separation, as in the water-oil system. In a simplest case, if we have a system with a solvent molecule and polymer molecule, and if we make a concentration of polymer higher, then at some point there will happen phase-phase separation with a phase with a higher concentration of polymer and another phase with a lower concentration of the polymer. And if we continue to increase concentration of polymer, then this two-phase system will become one-phase system again. And this phase separation process is sensitive to different variables of environment. For example, temperature, rising temperature, we could make two-phase system into one-phase system. And talking about functions of IDRs and biomolecular condensate, they perform a variety of functions. But it is interesting that in case of IDR, they are primarily related to the processing of intracellular information. For example, epigenetic regulation, GPCR regulation, it's a special class of receptors, cell signaling, in a more general case, genome maintenance, transcription regulation, and so on. And many functions of biomolecular condensates are also related to information. They can act as filters of some molecules as sensors or as resistors or buffers. My intermediate conclusion is that although the functioning of IDRs and biomolecular condensate is usually associated with weak interaction, a core role of IDRs is in the reception, processing, and transmission of cellular information, that is molecular communication. It's a citation. Everything that I have said so far is pretty problematic, is pretty well known. But as far as I know, nobody has tried to interpret this new phenomena in molecular biology in the context of philosophical discussion of biological information, and I will try to do it. I will try to show that this new phenomena problematize the concept of biological information, namely the concept of semantic information. Why I think so, and why I have highlighted that this tension between weak interactions and role in informational processes. One reason is history of science. Some time ago, biologists managed without the concept of information. For example, in her book, who wrote the book of life, Lily Kay argues that this dominance of information discourse in biology since the mid-20th century is linked to the epistemic rupture and partially could be attributed to the overall informatization and cybernetization of science and society during the World War II and the Cold War. Now I don't have time to talk about it more in detail. But the immediate predecessor of the concept of information was specificity as stereocomplementarity or as stereospecificity. I have already shown you pictures where proteins and other molecules have interacted as lock and key. And this lock and key hypothesis of enzyme substrate recognition is the oldest and the most famous example of concept of specificity as stereocomplementarity. There are more modern concepts of enzyme mechanism, for example, induced feed, but they are also in the same way. They also use stereo complementarity. Okay, and this stereo complementary interaction is supposed to be highly specific. By specificity of molecular interaction the specificity is usually defined as the difference in binding strength or affinity with different ligands. To some ligands this affinity is high, to some is low, and some interactions are highly specific, some are low specific. It is often assumed that binding strength corresponds to specificity, and that's why many authors talk about not, for example, weakly specific interaction, but they talk about weakly weak interaction, simply weak interaction. But what is interesting that before biologists decided to use stereo complementarity as a major concept, there were discussions and there were alternatives to strong specificity. And this could be illustrated by history of immunology. There was a debate in the beginning of the 20th century about the mechanisms of antigen-antibody interaction. According to Ehrlich, this antigen-antibody interaction is stereospecific, is not highly but even absolutely specific. But there were alternatives. There was a colloidal approach, there was approach of physical chemistry, and in both approaches, interactions between antibody and antigen were realized through weak interactions. And these alternatives to stereo complementarity, they have lost due to the development of structural chemistry, when it became possible to show how amino acid sequence defines 3D structure of proteins, and how this how proteins interact with ligands. But these colloidal and physical-chemical approaches, they were real alternatives, and they could be considered as unrealized alternatives. After this brief excursion to the history of science suggests that the information approach and the concept of weak specificity are radical alternatives, because they are because they were used in very different times. But I will mostly analyze contradiction between new phenomena and the concept of information using biological texts. Mostly the major review of function of intrinsically disordered protein regions by Kohlhaas and Kragelund, and I have already cited this review. And my hypothesis is that ideas and biomolecular condensate violate some intuitions about teleosemantic information, about some properties of teleosemantic information. These properties are italicized here. The first citation is not from this review, it is from the textbook of Alberts. And Alberts and co-authors write that intracellular signals must be specific and robust in a noisy cytoplasm. They also highlight the role of high affinity, high binding strength, and specificity of interaction between intracellular signal molecules and their partners. And they even write about complementarity. So they are talking about the high specificity as stereo complementarity. So the intuition is that only highly specific interactions can have informational properties. And if we read the review by Holhaus and Kragelund, we can see some disturbing thoughts, some reservations. They write that it is tempting to consider binding affinity as a proxy for the importance of the given interaction. But sensitive and responsive regulation of high affinity interactions raises many challenges, and weaker binding affinity may reflect interactions that are most easily regulated. So we can see a change of approach. Also, they highlight the role of context, and they understand that this low specificity interaction could lead to aberrant interactions. So it is not evident which interactions are important, which are not, which are informational and which are not informational, or even misinformational, could lead to miscommunication. These citations also could be used to discuss intuitions about functionality. The word importance is a proxy for a function in a biological context. So the intuitions seems to be like this: specific functions are provided by specific structures. What does it mean from a philosophical point of view? I will try to interpret. For example, it could mean that the function is provided by a mechanism that is at the molecular level is similar to a machine. So it's a machine conception of a cell. It's a mechanicism. Another interpretation, an organism or a cell could be reduced to a set of functional features, so it's a reductionism. There could be, I guess, another suggestions. The third intuition, the intuition of the sender and receiver of a message, they must have a certain structure, but at the same time, if you look at biomolecular condensate, they have variable composition, dynamic boundaries, variable size, and it is unknown at what size a biomolecular condensate begins to function and how a change in its size affects its functioning. So it's this notion of a sender and receiver of message, they became very fuzzy. And the fourth, intuition. This very interesting citation. Authors of the review, Holthaus and Pragelund, write that it is not appropriate anymore to describe proteins using a single interpretable descriptor, for example, like a kinase, the hydrogenase, immunoglobulin domain, and etc. These descriptors they contain information about function and sometimes about structure. And it is not possible anymore because ideas are conformationally heterogeneous and they are multifunctional, and their function is context dependent. So we have to embrace the underlying biochemistry and biophysics of ideas and do not use this this classification, this classical classification of proteins. And this means, as I think, this problematizes intuition of arbitrariness. According to this intuition, non-adjacent links in the causal chain connecting the alleged source and signal must be independent, but it is not evident anymore because these links, these proteins, they are multifunctional and their function depends on a context. So they could influence each other. For example, if links of the causal chain are inside one biomolecular condensate, then this environment will influence all links in the chain. And we can't anymore talk about arbitrariness in this sense. So my conclusion is that this new phenomena violate for at least for intuitions about semantic information, for different reasons, and what to do with this, how to understand this situation, this contradiction. I would like to describe some approaches which look interesting, namely approach of soft metaphysics and the theory of complex systems. And I have very little time, so I'll be very brief. The concept of weak specificity, the concept of causal emergence, and the concept of causal specificity. It is natural to consider ideas and biomolecular condensate as examples of biological soft matter. Soft matter combines viscous and elastic elements, so it combines properties of liquids and solid bodies. And which is interesting, soft matter exhibit large responses to moderate stimuli. And authors of the paper, which I use, Turoverov et alia, they suggest to combine approach of soft metaphysics with approach of complex systems theory, namely chaos theory, edge of chaos systems also can reply, also are capable of large and right responses to minimal changes in their environment. And I think that it is interesting not only apply to complex system theory, which many authors do. For example, Dan Nicholson, when he tries to understand how could be machine conception of a cell, how it could be changed, by what it could be replaced. He talks about non-equilibrium thermodynamics, complexity theory, self-organization, and so on. But I think that connection with soft metaphysics makes this approach more concrete, more interesting. And of course, if this approach will be successful, it will explain many things. It will explain how ideas and biomolecular condensate could function, despite that they are not machines, and despite their holism. It could clarify dynamic character of this new phenomena as senders and receivers of information, could reveal the non-arbitrary nature of correspondence between sources and signals. But as far as I understand, the success of these theories in this field is limited. Another approach is from a history of science, as I have already described. The weak specificity. I think that this framework could provide a common name for this new phenomena in molecular biology. Not only ideas and biomolecular condensates, but also enzyme promiscuity, molecular crowding, and there are other similar-looking phenomena which I think what they have in common, they are grounded on weak specificity. Maybe it's a trivial name, but I think that it is neutral, and that's why it is useful. And also, this concept of weak specificity could a bit clarify how the idea function is determined. It is determined by a combination of short linear motifs, ensemble properties, and sequence features. And the first is about strong specificity. Sequence properties, features, they are about weak specificity. And I think that ensemble properties, it is also could be conceptualized as determined by weak specificity, by weak interaction between many assets in the same protein. Two other approaches, causal. The first is causal emergence and causal spreading. Causations could be quantified, and some other authors say all metrics of causation show that in complex hierarchical system, causation is largely localized at the upper levels, and it is called causal emergence. For example, in one study, such causality measure as effective information was applied to protein interactomes for many thousands of species, and it was shown that in these interactomes could be found informative macroscales when clusters of weakly specific protein-protein interactions, which interact closely to all mapping, can be replaced by individual nodes, and all this without losing informativity in the network. And according to this study, causal emergence is more pronounced in eukaryotic protein interactomes, not in prokaryotic. And this notion of causal emergence, of course, could clarify strong dependence of ideas on biomolecular condensates on the context. Very thoughtful discussion about this causal emergence is provided by Philip Ball in his papers and his book How Life Works. And here he suggests to talk about causal spreading, not causal emergence, because a gain of causality at the higher levels is accompanied by the loss of causality at lower levels. So it looks like a causal spreading. And the last approach is a usage of information as a measure of causal specificity. This approach is based on a manipulationist concept of causality. And here information is understood as a measure of the detail of possible intervention and control based on knowledge of causal relationships. And the measure of causal specificity is the mutual information between manipulations of the independent variable and the values of the dependent variable. The more fine-grained is control obtained by manipulation of the independent variable, the more is causal specificity, and the more information. Which is interesting. This measure could be used for continuous cases, which is important for weak interactions, and I think that it could clarify the property of non-ubiquity of information, because it proposes a quantitative measure instead of yes or no relationship. In these two last approaches of causal emergence and information as a measure of causal specificity, they look compatible, and I think that it is possible to use information as a measure of causal specificity to quantify causal emergence. So my intermediate conclusion is that these four approaches, they are the last two are somewhat similar, but they are different from others. One is from science, the other is from history of science, the third and the fourth are from philosophy, but they could clarify some intuitions. And my general conclusion is that the concept of information is embedded in the discourse of molecular biology, typically is interpreted semantically. However, this interpretation is difficult to naturalize. And new phenomena in molecular biology, they make this interpretation even more difficult, because they problematize such properties of semantic information as non-ubiquity, naturality, and the presence of either arbitrariness or a sender and receiver of a message. And I think that in order to understand this phenomena in the informational context, it may be useful to turn not only to philosophy, but to some other science and to the history of science. Thank you for your attention.
We have a 5-minute break. And we're back at 3:10.
Okay, yeah. Questions?
Thank you so much for the talk. It was super interesting, super dense. I really like this project. I just have some maybe clarification question. So let's try to. So you said that. So the biologists, even if these very present biological textbooks, are very engaged with this idea of information and in terms of telesemantic information, right? This is the important point. And you underline that.
Yes. Yes.
I
But
comprehend.
there are other interpretations of information, and but they are less known.
Okay.
Yes.
Your interpretation of the information.
I suppose that biologists are using telesemantic interpretation.
This is,
Yes.
This was the.
Yes.
Okay.
Without knowing it.
Okay, yes.
But
You can that's a philosophical interpretation of
But they
some.
probably think they use Shannon, but they didn't check.
Yes, but it's not Shannon. Yes.
And so my question is, so and you propose to point at some problems concerning these semantic informations, because we have this new phenomena and it's very hard to frame this new phenomena with this standard view.
Yes.
Okay, this is only for me. And my point is, because nonetheless, telesemantic information is related to this problematic notion of function, right? Telesemantic information for you, for your interpretation, for your account, is something that enables biology to says something about function, okay? And but in biology we have because in philosophy we have a problem concerning what is function, the more evolution one, the systematic, etc. This is a philosophical problem concerning function. But we also have a very contemporary biological problem concerning functions. For example, there is some struggle in understanding which proteins can be functional, which is just a molecular error, for example. So we have a problem in the very biological framework concerning function. And so my question for you is, sorry to that, but I love this, so for you, so do you think that your account can also help to set this biological problem besides all the philosophical debate problems or stuff that happens? So your idea that we can think about weak selection would be useful also for biologists themselves in order to be able to understand better what is a function in molecular domain. This is the question. Thank you. I have another question.
Yes, I think that this notion of weak specificity is underused. For example, biologists write about either specific interactions or about lack of specificity. And this intermediate notion of lack specificity, it looks strange for biologists, how it is, what to do with this. And yes, I think that it is useful to think in these terms. And approaches which I have described, they could be useful not only in the context of discussions about information. They could clarify conception of function overall, yes, and not necessarily alluding to evolution. I was talking about etiological concept, function, which defines function through natural selection, and it is useful in informational context in order to explain how intentionality arises, but we could use another notions of function without thinking about history or about evolution in this context. And I think that these approaches, they could be useful in these cases too. Yes.
Thank you.
So I'm a bit. I'm a bit obsessed by informational concepts, and I look through these lenses, but actually approaches which I'm talking about, they could be useful in other contexts too. And this my obsession, it contrasts my approach from Dan Nicholson approach, who sees a cell as a machine, or to be precise, he criticizes this approach, and he thinks that this machine metaphor is central. And to my intuition, informational metaphor is central to molecular biology.
Yeah, thanks. That was really interesting. I have a question about, which also is what caught my eye in the abstract, which was the aspect about history of science. So, I'm trying to understand what you mean by that there's a problem to naturalize the concept of information, and so I want to understand what you mean by naturalize, and why is it a problem that it's difficult to naturalize, and whether I feel like your project is to be able to naturalize
Yes.
that, right? And so so you nicely showed how in different periods in history, different metaphors, different notions are more prominent. We had the information sort of cybernetic metaphors that are still very pervasive. There was before the sort of more physical, chemical approaches, and that different sort of concepts, different metaphors allow for certain phenomena to be explained or better or worse, like the weak specificity and so on, the condensates at the end. And so you said very nicely that those are sort of different alternatives. So things could have been otherwise, right? We could have now something beyond information that's sort of somehow culturally dependent or culturally contingent.
Yeah.
And so I'm wondering how that vision of history where you have different alternatives helps you in your project to naturalize information. I'm just not sure. I don't know if that question makes sense, right? So it feels like you still need some sort of normative work to say, well, it's better information to do this kind of stuff, and it's better the physical chemical metaphors to do that kind of stuff. And I'm wondering how history of science can help without being normative, and if you are being normative, in what way?
Please explain one more time about normativity.
Right. So if you look at the history of science, like you've done, you have different alternatives.
Yes.
For how to conceptualize different biological phenomena.
Yes.
How does that help you naturalize information?
It helps me to problematize, to see. When I was citing biologists, we could see that they are very quickly turning from contradictions to some perspectives. They are ready to forget their old intuitions and to construct some new intuitions. But and it could look that everything is clear now, but it is not true. And I was comparing textbook, modern textbook, and this Nature Review, and there are contradictions. And in order to know these contradictions, in order to problematize, I use history of science, to show indeed that there could be alternatives. They were already realized in different contexts, but they were. And methodologically, I very like this paper by Lazon, and I think that it is the most thoughtful discussions about the problem of presentism in history of science. Approach which I used could be blamed by historians of science as presentist approach. They could say that it is not appropriate to use history of science in modern discussions because it was absolutely another context, and history of science should be explored for its own sake. And that this presentism position is to study history for the sake of the modernity. This position indeed could lead to distortions, to problems, but there are different forms of presentisms actually, and if we think about them, we could use history of science.
But just very short, you use history of science to problematize information, right?
To problematize modern science and modern philosophy of science.
Because you don't think that the project to naturalize information is even possible.
And I think it is possible, but there could be more obstacles than philosophy for science think.
Okay. Yeah, okay. That makes sense. Thanks.
Thank you very much for the talk. I didn't know about the IDRs, but I think it's a very interesting case, and it's pretty convincing. So, semantic information has a problem there. But so my question are all about your approaches to solve the problem. And I'll ask one, and after that I can come back with I have many. So first one, I didn't understand well the difference between causal emergence and causal spreading. What is the difference in the strategy? I'm not sure I understood.
The notion of causal spreading highlights that the gain of causation at high levels is accompanied by the loss of causation at low levels. At molecular levels, when there was a transition from prokaryotes to eukaryotes, at molecular levels, eukaryotes are more noisy than prokaryotes, but they regain causality and specificity at higher levels.
So isn't it a specific case of causal emergence, a special case, or?
Yes, I think
Oh,
yes,
okay,
but
okay,
it's
So it's why you put them together.
yes, and this notion of causal emergence is of course more well known, but there could be another name, I suppose.
I have others, but I don't want to take
A question myself? Yeah, so my question is about the very beginning, where you said that there is information and it becomes problematic when we interpret it ontologically, explanatorily, and literally and not fictionally. And I was just thinking how this claim relates to where you end up in the end. So in the end, given that, for example, this new phenomenon, the fact that even naturalized semantic notion is not doing well with them and we need something else, with this something else, we still cannot interpret it ontologically and explanatorily, or if we invent some clever naturalization, we will be able to claim
Okay, so one alternative is to reject the at least the semantic interpretation, and I think that some approaches are indeed they look as epistemological approaches, but some could be ontologized. For example, causal specificity. Maybe it could be ontologized, and we could say that it really exists in biological systems.
others, but
go on.
Okay, it's about the first one, the soft matter approach. I'm not sure I'm following you here, because even if a soft matter model with some chaos for fun to have some nonlinear effect managed to fit biology, or at least certain biological system, I don't know how it would be explanatory at all. Because it seems to me that what you're looking for in all the cases you discuss, it's pretty mechanistic causal explanation in biology. Of course, the kind of explanation you have for system dynamics, it's at such a high level of abstraction. So I don't know if it will answer anything that you want for. So you seem to be more optimistic than me, so could you explain why? But you said it was lacking, or not? No, you didn't say lacking. You said limited. Limited. You used the word limited. Capacity of explanation.
Yes, I think that this approach is an alternative to mechanicism, and
so just to illustrate, I bring you a bunch of thermodynamic equations. They all work together. There's some causal blah blah blah. It's closed blah blah blah. And I say, aha! I explain everything you need about cells. And you would as a biologist, you would say, really? You fit something, you build something that is cool, but is it explanatory?
To me, actually, yes.
Okay. So what part?
So I
What
think
part
that
are you optimistic about?
and I'm suspicious of mechanicism in molecular biology, and I think it's so there are authors that they're talking about new mechanicism in molecular biology, that they think that it's a special feature of molecular biology, that there is no theory. There are these pictures of causal relationship, but I think that this situation will change. It is already changing, and this soft matter concept is a right way to describe. Maybe not all, but many phenomena inside the cell.
But we can agree it's a good way to describe, but is it a good way to produce explanations?
I think that if I'm not used to, I think that good explanations are connected to predictions. And that's what I'm waiting for.
So if it's able to predict.
Yes, when it will be predictive. And I will show you one picture about this approach of this picture. From Turoverov and et al. Paper. This paper is by authors that have described the importance of intrinsically disordered proteins. Some of these people were at the beginning of the research of these proteins, and they see and they build analogies between systems, between the edge house systems and ideas. Here you could see some classical edge of house system, Lorentz attractor, and dynamics of its variables are very, or not very, it's hard to see, they are similar to dynamics of ideas. In this graphic, so-called Fred efficiency is a measure of compactification of a region. The more efficiency, the more compact is this region. And we could see that there are, as the authors suggest, these intrinsically disordered regions stochastically switches between more and less compact information. And this is, as far as I understand, this is the level of the level of the degree of connection between these physical theories and these new
free
phenomena.
variables in their model? How many free variables to fit the data?
Yes.
Is there 2, 3, 4, 10, 15?
Yes. So it's only some analogies to this problem.
Because I don't want to insist, but we had the same problem, because I'm not a philosopher of biology, but in sociophysics, you can fit some phenomena of voting with thermodynamics, and it's pretty predictive and very efficient. But you turn to sociologists and they say, okay, that's cool, but that's not explained. We need causal explanation. We don't want to just your fit mathematics. Of course, we learn something by fitting the mathematics. I agree. It's not by complete randomness that they fit. But would you provide explanation that will satisfy a molecular biologist? To a question why. So if the molecular biologist, why that? And you say, Because I worry between the explanatory power of a model and the quality of prediction of a model, the fitting of the model.
And I think that biologists are too obsessed with these pictures of mechanisms. I think that it is a
That's not our fault. That's
yes.
They do their things.
Yes,
Okay, maybe they are too obsessed now about the chemistry.
Even in history of science, this stereocomplementarity approach, which is so successful in molecular biology, was even called a morphological approach, and it was contrasted to physical-chemical approach. So it is approach not of molecular biology. It comes from, we could say, from zoology. This notion of complementarity of structures, this thinking in terms of fixed structures that complement or do not complement each other. And I think that this situation will change. And these predictive models, they will come to biology, and they are already coming. We have to embrace this biochemistry and biophysics as the authors of the major review, right? Biologists have to deal with it.
I have more. Sharon, maybe.
So concerning the two new phenomena that you discuss, the molecular condensate and intrinsical disorder proteins, and so you said that we need, from these new phenomena, we need a new approach. But first of all, I would like to, I don't know if you show us this part, but how, because concerning these specific two new phenomena, I'm struggling to see the difference between information and functions. Because it's very clear for me the very three cases in which whatever biologists use information, the central dogma, the code, the intracellular signaling. But what is the perception by biologists concerning this? Because it seems to me that here they work more in a narrative in which they use function than information. Because you, for example, mentioned different function of intrinsic disorder protein and biomolecular condensate. And here, maybe I confuse a bit concerning where information here is present and we should reframe it. Or maybe I'm just confusing the pieces. Can you give a clarification about that? Because maybe it's me.
I think that biologists used the same criteria that philosophers are discussing: arbitrariness of correspondence between an enlarged signal and an enlarged source, and the presence of a receiver and maybe a sender of an information.
Okay.
But these notions are fuzzy. It's a problem.
Yeah, because for me here is more about function and not information. But if you frame information in your categories, maybe I can understand the link between the two. But it's not so intuitive for me. But it's intuition. It's not. I'm not studying this. So it's very clearly function, but. So you're saying that in light of the fact they are using this category of sender, receiver, they are embracing an informational framework, right? This is your point? Okay. Okay, I see.
Biologists very like pictures. For example, in these pictures we have some clear sender of information receptor on the cell membrane, and we have some clear receivers of information, and these links in this causal chain, they could be changed, as is written in a textbook by Alberts et al. In a cell, any molecule could influence any other molecule because of modularity, because of independent evolution of different models of these causal chains. And if we see this modularity, and if we see some defined structures which could be interpreted as senders and receivers of information, then there is a biologists talk about information. And that I think that their intuition is caught by philosophers rightly, correctly.
Okay. Sorry. Could you explain more the difference between weak specificity and lack of specificity? Because you want to bring back weak, and I would like to better understand your concept.
I think that biologists talk about lack of specificity when they do not want to acknowledge weak specificity. So they talk about this weak specificity like this is something abnormal, and they call it lack of specificity. But there are really some examples where interactions are very, very low specific. I will show you an example. Here, interaction between some protein, for example, IDR, and crowding agents. Cell, there is a very little free water in the cell. Cell is very packed by different molecules. And interaction between a protein in these many different molecules, they could be averaged as non-specific interaction. So I think that this is the closest example of the lack of specificity. But even in case of crowding agents, some crowding agents, different crowding agents, they interact differently with protein. And when biologists trying to explore this phenomenon, they have to very carefully define a crowding agent, what crowding agent to use, because for different proteins, the same crowding agents could lead to very different results. So actually, it is also not a lack of specificity. There is some specificity, but very, very low.
Thank you.
Follow-up concerning weak specificity. Apparently, that biologists already use weak specificity, right? In some specific example, as the very famous weak in the DNA, for example, the hydrogen bond, etc. So they use already this narrative of weak specificity, right? What is how you frame your proposal concerning the fact that nonetheless biologists still use this weak idea of specificity?
In case of DNA, biologists suppose that many weak interactions they sum to a strong and highly specific interaction. So in the end, they deal with a highly specific interaction.
So weak bound.
Yes.
Bound the high specificity.
Yes, many weak bonds lead to high specificity. But in case of ideas and biomolecular condensate, this is not the case. These weak interactions, weakly specific interactions, they are very important by themselves.
I have a couple of questions. So the first one is regarding the machine metaphor. You said that you prefer to use information as a central concept for molecular biology, but still, indeed, machine metaphor is clearly present there. And I was just curious, what's the impact of specifically these two phenomena on machine metaphor, whether it's completely disrupted or we can still preserve it, but now it will be soft stochastic machine, but still it will be something spatially localized, or
Nicholson thinks that this new phenomena and some other phenomena, they destroy this machine conception. But biologists tend to think that the old notions and the new notions, they are complementary. They are about different processes or different parts of the cell. For example, in the textbooks, these biomolecular condensate, they are contrasted to all the notions of macromolecular machines, membrane enclosed compartments, and the authors try to show that they are complementary, and this could be done for ideas too. I'll show you some picture. This is the picture. Actually, it's this picture could be interpreted both radically and both as a compromise. The radical thesis is that proteins usually consist of regions with different conformational stability, and their structure is mosaic. So it is not the case that there are some classical stable proteins and there are proteins with IDRs. All proteins they have mosaic structure, and this leads to, this is another term that biologists use, not weak specificity but binding promiscuity. And it is also a negatively charged word, like lack of specificity, even more pejorative, promiscuity, binding promiscuity. So biologists don't like this phenomenon. But these different regions, they could be ascribed to different processes. So according to this classification, which is by Uversky and other authors, there are classical fold-ons that are independently folding structural elements, and they are used in transport and catalysis in the cell. There are inducible fold-ons that take a form when binding, and they are used for molecular recognition. There are non-fold-ons. They are used for entropic chain activities. They just move in the solution, and they they move away all the structures. They are nearby. They just move away from everything. There are semifolders which are always partially folded. They're used in signaling, and there are unfolders, structured elements that must unfold to perform a function. So the bad news for biologists is that protein seems to be mosaic. They have a mosaic structures, all the proteins. The good news that elements of this mosaic could be described to different functions. And biologists prefer to think that this new phenomena, they could find some place for this new phenomenon, not to make a revolution in molecular biology.
Can I ask, so why is it a bad news for biologists? It's biologists who found that, so I'm just, why is it bad news?
Because biology is getting more and more complicated since its beginnings, and there is no end to this process.
That's good news, they're going to get employment forever.
But it is very hard to understand this phenomenon. So it is hard to make sense of all this data. It is overwhelming, and biologists write about it. So there is no end to this complication. There were very big hopes about human genome project, but this project only shows that biology is more complicated than was thought before. And all these promises of new drugs and so on, they are mostly not fulfilled, these promises. There are only new dimensions of complexity. So there are bad news. The biologists still don't see the end of these complications.
You don't want just to be a science that put names on things.
Yes.
You want to work.
Yeah, that's the question that you asked about.
But yeah,
Explanation, it's very relevant.
yeah, they want to provide explanation, so just that. Because it's too complicated. They want to provide explanation, general theory. There's a biologist here, but he said nothing.
I put to myself. Oh no. Ask your questions.
So we touched a little bit the topic concerning stochasticity, and I try to avoid this but at the end of stochasticity on the left, stochasticity on the right. So, how can you frame the stochasticity in the cell concerning this idea of function? What do you think? So it's very a curiosity because you said not specificity, and because we are crowded cells, and sometimes in the average it seems that we have no specificity. Are you saying that we have stochasticity? And if yes, what is the place of stochasticity on your framework?
I think that it is very important, and the usage of this physical theories I was talking about could help to work with stochasticity. But conceptually, I like the point of view of Philip Ball, In eukaryotes, stochasticity on a molecular level is increasing, and it also means that genes are not that not that causally important in eukaryotes, and that's why many mutations do not have some important consequences in eukaryotes, but the specificity and causality and determinism is transferred to higher levels of organization. It is not necessarily. I'm sorry, I have to show this slide. Not necessarily we are talking about transfers of causality from, for example, from molecular level to organelle level, or from molecular level to cell level. There could be intermediate levels of causality, some subcellular networks that are robust. Biologists prefer to talk not about determinism but about robustness. They are robust, but their components on the molecular level they are noisy, they are stochastic, and I think that it is really the difference between the elephant and the E. coli. Like Philip Ball writes, he is referring to the famous citation by the famous phrase by Jacques Monod, who said that what is true for E. coli, for Escherichia coli, it is true for the elephant from the biochemistry point of view. But now we see that there is a difference. These ideas in biomolecular condensate, for example, they are common in eukaryotes and they are rare in prokaryotes. So we see that stochasticity increases at molecular levels in eukaryotes compared to prokaryotes. But this is complemented by increasing of robustness on cellular level, subcellular levels. I like this picture. And it is also one more way to find some place for this new phenomenon. Not to say that they are overturning that the old paradigm of molecular biology. We can find them place, for example, comparing prokaryotes and eukaryotes, or comparing their role in different processes.
It's a complementary. But I'm surprised that you're not saying something about your fourth approach. See, if you understand information as a measure of causal specificity in a manipulative account, a causal link in a manipulative account, it's a dependence between stochastic variables. So it seems that you are connecting information with certain dependence of stochastic variables. If you adopt the fourth approach, it's maybe indirect, but it's still there, because the manipulative account is a measure of dependence between stochastic variables, between variables that most of the time are stochastic, because you're in biology and nothing is deterministic.
Could you explain?
Because if you say, okay, information as a measure of causal specificity, I don't know if my notes are okay of the fourth approach.
Yes.
Yeah, information as a measure of causal specificity, and you use the manipulative account.
Yes.
Okay, so the manipulative account is saying if I do something here, I have something here. But that's the way we explain it. But of course, when we model that, it's if I had changed the dependence of this stochastic variables, I would have had a change of the distribution of probability of the other variables. That's the way the mathematician understands the manipulative account. And of course, that's the real definition that is defined by Woodward, by Pearl, that works all the time, even in deterministic systems. These are just special case. So if you say, my notion of information is a measure of the strength of this connection between stochastic variables, it means that for you, the notion of information is related to dependence in a stochastic system.
So
At least it's the way the definition is said. So you seems to have in this proposition of information that seems better than the previous one, because it's naturalized. That's a naturalized proposition for Max. That's a naturalized proposition.
yes.
It's already including implicitly some stochastic aspect. It's just that information will not be stochasticity, it will be dependence between stochastic
Yes.
phenomena.
Yes.
So it's just a complementary to your question.
Yes.
And I was surprised that you didn't say that because you had that in your own approach.
Yes. Thank you for your addition. Thank you. I agree.
Sure.
That's it.
All right. Thank you so much.